Good afternoon, everyone. My name is Daniel Jiménez Martínez, and I will present the project Evaluation of Nanostructured Carbon Platforms for the development of urea enzymatic sensors, which was carried out in collaboration with and the advice of the PhDs Alejandra Álvarez, Sandra Rivas, Vanessa Vallejo, and Juan de Dios Galindo. Without further ado, let's begin. The aim of this project was to propose and evaluate an electrode capable of urea detection through the use of nanomaterials that could further be used to develop a highly selective and a specific biosensor. But now, what is a biosensor? Well, biosensors are analytical devices. These convert a biological response into an electrical signal. For the last decades, their development has been of great interest on the research field due to their unique high selectivity and sensitivity properties. As we can see here on the presentation, I show you a typical design of an enzymatic biological sensor. Which is the difference? Well, for an enzymatic biosensor, we need as a biological recognition molecule, an enzyme, which is highly specific and, well, which can help us study and analyze. And according to the transducer, we can have different types of biosensors, which are known as piezoelectric, thermal, optical, but the one we're going to be focusing on is the electrochemical biosensor. Electrochemical biosensors have an electrode, which is the thing that we are proposing for this project as transducer. This offers so many advantages as high selectivity and specificity because of the main constituents of an electrochemical biosensor. Now, what do we want to analyze? As I mentioned you in the title, we want to analyze urea. Urea is of course of great importance. We want to analyze it because of the problems it can carry out in the dairy industry as well as the clinical industry. Urea detection is of great importance because blood urea nitrogen has been utilized to evaluate the renal function for human beings, and of course to know about some diseases. But it can also be applied, as I told you, in the animal care. In example, it has been reported that when the milk urea nitrogen reaches more than approximately 20 milligrams per deciliter in cows, it can cause some problems on these animals. But of course, the greater importance yields here in urea detection for biomedical applications. Why? In humans, urea levels serve as biomarkers for kidney problems and obstruction of the urinary tract. What happens? High concentrations may cause bleeding and dehydration. Urea is generated by the urea cycle enzymes, which are mainly in the liver, but are also expressed at low levels in other tissues. And as we can see, a change in the uric cycle can increase the ammonia levels. Ammonia is the product of the reaction of hydrolysis between urea and ureas. So we have our analyte, we have urea. Of course, if we want to analyze urea, what are we going to need as an enzyme? Well, we're going to use ureas, of course, because it causes the hydrolysis of urea, which it can produce ammonia. Ammonia is very important. Why? As I mentioned you, the ammonia levels increase within the problems that occur with the uric cycle changes. And of course, with production of ammonia, we can make an identification. But how are we going to do this? How are we going to create this biosensor? Well, first and foremost, I need to talk to you about another type of things. For example, enzyme immobilization. There are many types of enzyme immobilization. For example, we have physical immobilization and chemical immobilization. We're looking forward to make a chemical immobilization. Why? Because physical are weaker. They are weaker, and suddenly our analyte and the enzyme may separate from these things. And of course, with the chemical immobilization, we include weak bonds and covalent bonds. In this project, we're going to be analyzing 3 different carbon nanostructures, which are going to carry different types of bonds. For example, we're going to use carbon nanofibers, we're going to use bulk and carbon, and we're going to use carbon nanotubes, multi-wall carbon nanotubes. These will exhibit different types of immobilization, but of course, our main interest to use them is their properties, their high electrochemical properties, such as, well, high nanometer size, and the volume radio of the speedway that it can analyze and convert signals. But I mentioned you before, we're going to be working with both types of immobilization. Why? Weak immobilization is due to absorption or even weaker bonds. And with covalent immobilization, as its name implies, we're using covalent bonds. With multi-wall carbon nanotubes, we're going to be performing a covalent immobilization. And with the other two structures, we're going to be performing a weaker immobilization, which is the weak bonds immobilization, absorption mainly. For our objectives, I am going to present you, well, in a general way. We want to create these sensors, we want to evaluate it, and we want to compare different carbon nanostructures. And of course, with the specific objectives, we're going to check out which one performs better at different analysis. For the methodology, the first part was the oxidation of ammonia, which is the main product of the hydrolysis of urea. How are we going to do this? Well, through some research, we found out that iridium can oxidize ammonia. For that reason, we're going to be proposing to use iridium dioxide nanoparticles, which exhibit so many good properties for urea identification. And of course, well, I'm not here to tell you, you may not be able to hear, but urea cannot be determined directly electrochemically. So we're going to have to use something else, which in this case is going to be the iridium dioxide nanoparticles for the ammonia oxidation, for it to take place and of course be afterwards analyzed to identify urea. Now, the synthesis of iridium dioxide nanoparticles. We followed, well, we based our research and our synthesis in the work of Chakrabhani and collaborators from 2015. But we changed the stabilizer agent. For a reduction synthesis, we need so many things. We need an stabilizer agent, which in this case is ethylene glycol. Secondly, we need, of course, the material that we are going to be synthesizing. And afterwards, we need a reducing agent, which is in this case the sodium borohydride. Now, once we have our nanoparticles, in a few minutes, I will show you how to characterize them and what we did with them. We prepare the catalytic ink and of course our electrodes. We made a catalytic ink using our nanoparticles, our nanostructure carbons, platforms, and of course we added the enzyme for it to be immobilized. And oh, again, we have iridium dioxide nanoparticles as the main protagonist here. Why? Because as we can see here in the reactions, we have urea plus water, which is the hydrolysis. Through urea as a catalyst, we are going to be generating ammonia products. And the interaction of the iridium nanoparticles with ammonia produces the oxidation of this molecule, which can be analyzed. But how it can be analyzed? Well, of course, here I present you with the instruments that we use. I'm going to go further to be faster. And now I'm going to show you the results. First and foremost, we need to characterize the nanoparticles. The nanoparticles were mainly characterized by scanning electron microscopy. Why? To grant that these are in fact nanomaterials. Then we proved with the free enzyme stability. Here we added nothing. We just wanted to see which are the best conditions in which the enzyme can produce ammonia. So we used urea solutions, ureas, and of course we measured it with residual activity. We found out that between a pH from 6.25 to 7.25 is the best pH on which we can determine this type of analysis, as well as temperature. Now, for the enzyme coupling, here we want to see how much of the enzyme is in fact going to be added to the material, because we may say, oh yes, yeah, I'm going to put my enzyme, but is it true that the 100% of the enzyme is going to be on the final product? Well, it is not true. So this is what we did, an enzyme coupling. And as we can see, this is the first part where we can make a comparison between the three nanostructures. Why? Because the Vulcan nanostructure exhibits the best ammonia coupling, including the best enzyme coupling, which is ureas. And then let's go to the most important part, electrochemical sensing. Here we're going to see if our materials really make something for urea identification. As we can see here on the figure 14, a cyclic voltammetry was carried of a Vulcan electrode with, of course, our material and ureas against a glassy carbon curve, which is the one used from the working electrode. And as we can see with the red red graphic, with the red line, there is in fact an identification. There is happening an oxidation reaction which creates that curve, which allows us to say, okay, we're producing ammonia, which in fact means there is urea present in the solution. But of course, more work has to be done, and I'm going to show you right now. Let's try to compare the three different carbon nanostructures, starting with Vulcan. Here, as we can see, we present a curve again of cyclic voltammetry of the electrode against different concentrations. As we can see, here it was different. The blue line indicates that the iridium dioxide nanoparticles with the ureas are in fact the main point of this project. Why? In the red line, we can see that there is no side signal, but after we add ureas, the enzyme, and the iridium dioxide nanoparticles, this one can be carried out, as well as this one. We performed this analysis for the three carbon nanostructures. For questions of time, I am going to go faster through these ones. Let me show you. Here is the carbon nanofibers. And as you can see here, I am going to compare those three curves, but now only choosing the electrodes with the different nanostructures. And as we can see, the carbon nanofibers exhibit the best sensitivity. Now, what are we going to do with these things? Where we are going to create a calibration curve. How are we going to do it? Well, first we start with a differential pulse voltammetry of the different proposed electrodes to take a look at the currents and how it changes, to propose if it is going to be an amperometric or a potentiometric biosensor. Here, as we can see, the results are carried out, and with increase of the concentration, also there is an increase for the current given by the analysis. Here is clearer with this cyclic voltammetry. You can see perfectly how it changes with the concentration. If we add more urea concentration, then the current is going to go higher, even higher. Then what happens? Well, the peak values that we can see here on our graphs are required to design the calibration curves which are shown here. And this one, well, as we can see, these ones are stable and they have good numbers. And with these ones, we were able to identify the detection limit, which is 0.433 millimolar, which is far below the limits that we want to identify either in urine or in milk from the cows. These levels are far lower, which means that ureas can be identified. And with this one, we're going to create a sensing and of course, give you the current on which one on which these electrochemical biosensors will work. For the reuse test, we're going to ask us a question. Okay, yeah, I have my biosensor. Okay, you showed me. Okay, yeah, it's calibrated. It's good. It's okay. It can identify urea, but for how many times will I be able to use it? Well, it's very simple. And for that, we made a reuse test. With this one, we analyzed the different carbon nanostructures. As we can see, the multi-wall carbon nanotubes have the best activity through time. Why is this? Well, it's because of the enzyme immobilization. As I mentioned before, we're doing a covalent bonding with the enzyme and the iridium dioxide nanoparticles, which are decorated and as well immobilized on the carbon nanostructure. Finally, and as a conclusion, we can say that yes, the urea identification was carried out, but actually higher sensitivity doesn't mean that it's going to have better characteristics on the other analysis. One thing is sensibility, and the other one is stability through time. Therefore, even though the carbon nanofibers showed or exhibit a higher current and a higher sensitivity while doing the electrochemical analysis, they showed even lower or worse results through other tests, like the reuse test, and as well as the enzyme coupling. Well, no more for the moment. I thank you so much for your attention, and I'm looking forward to answer your questions. Thank you very much. Let me share the contact.
Muchas gracias, Oscar, por tu presentación.